The anatomy of the somatic motor system consists of neurons originating from the brain or ventral horn of the spine. When these neurons get close to their target skeletal muscle fiber, they branch out to control many muscle fibers at one time. The synapse between the neurons and muscle fibers in the neuromuscular junction. The neurotransmitters released are acetylcholine, and they are received onto nicotinic cholinergic receptors on the motor end plate. The anatomy of the sympathetic system consists of neurons originating from the first thoracic to second lumbar segments of the spinal cord. These preganglionic neurons then bind to ganglia in the paravertebral sympathetic chain. The neurons release acetylcholine onto the autonomic ganglion, which then creates a potential which travels to its varicosity axon terminus to release norepinephrine onto adrenergic receptors of target tissue, except in certain organs like adrenal medulla. The anatomy of the parasympathetic system consists of neurons originating from the midbrain, medulla, and second to fourth sacral segments of the spinal cord. These preganglionic neurons then bind to ganglia on or near the target organs. The neurons release acetylcholine onto the autonomic ganglion, which then creates a potential which travels to the varicosity axon terminus to release acetylcholine to muscarinic receptors of target tissue. Next, the adrenal medulla is in the adrenal gland, right above the kidneys. It is pyramidal shaped with two layers, the adrenal cortex and the adrenal medulla within the core. The adrenal medulla is a modified sympathetic ganglion and a neuroendocrine tissue that primarily secretes epinephrine. Adrenal medulla is activated by preganglionic sympathetic neurons that project from the spinal cord and synapse with the axonless postganglionic neuron, chromaffin, in the adrenal medulla. The neurons release acetylcholine, activating the chromaffin, which secrete large amounts of epinephrine directly into the blood for distribution generally throughout the body. This is an intermediate step in the fight or flight response. Next, the neuromuscular junction is a synapse between the somatic neurons and muscle fibers. This junction is covered by Schwann cells that form a thin layer covering the top of the axon terminal. Schwann cells help form and maintain the junction. The three main parts of the junction are the motor neuron's presynaptic axon terminal, the synaptic cleft, and the postsynaptic muscle fiber, which is formed into a motor end plate. Action potentials travel down the somatic neuron, depolarizing the axon terminal. This opens voltage-gated calcium channels into the presynaptic membrane. Calcium diffuses into the cell down its electrochemical gradient, triggering exocytosis of acetylcholine-containing vesicles. Acetylcholine diffuses across the synaptic cleft and binds to the nicotinic cholinergic receptor on the muscle fiber. The nicotinic cholinergic receptor is a channel protein that allows sodium ions to enter the muscle fiber and depolarize it. Many receptors are at the upper sections of the motor end plate gratings. To relax the muscle again, acetylcholine diffuses away from the receptor in the synaptic cleft, or is catalytically broken down by acetylcholinesterase.